Every living organism carries out chemical reactions in order to sustain life. For these chemical reactions to occur, every organism needs enzymes. Enzymes are in no way reactants or products in reactions, but rather they form an enzyme-substrate complex that then produces the product. The enzyme, after producing the product, can then attach to a new substrate and create more product.
Enzymes exist as a chain of amino acids, and it's these chains that are folded up to form the tertiary structure. These structures are what catalyze and make reactions run. We're aware that these reactions run at room temperature, but our experiment is to find out what would happen if these temperatures were to be changed.
Our hypothesis is that hotter temperatures will lead to a faster enzyme rate of activity, and colder temperatures will lead to a slower rate of enzymatic activity.
The enzyme we used was called polyphenol oxidase and is found in potatoes. To do our experiment, we were going to have to use potato extract. To get this extract, we took potatoes, put it in a blender, and ran the blender. This broke the cell membranes, releasing the cytoplasm where the enzymes contain, allowing us to experiment with it.
Polyphenol oxidase catalyzes catechol to form orthoquinone. This product produces a reddish-brown color, which then we can detect in a spectrophotometer. Using this, we can measure the absorbance and therefore find the rate of the enzyme.
We took 4 test tubes of 5 milliliters of buffered catechol each, and then added 0.3 milliliters of our extracted enzyme. These 4 test tubes were each ran at a different temperature. The temperatures included 0 degrees Celsius, obtained through an ice bath, 20 degrees Celsius, which is room temperature, and 45 degrees Celsius and 62 degrees Celsius, which we obtained through a heated water bath.
The spectrophotometer we used was the Milton Roy 20D. Prior to running the experiment, we cooled or heated the enzyme according to our guidelines, as well as the catechol. So when we mixed the catechol and enzyme, they were the same temperature. When we mixed the catechol and enzyme, we made sure to shake it to evenly distribute the enzyme. Before we put each spec tube into the spectrophotometer, we wiped it with chem wipes. In between each reading, we placed the spec tubes in its appropriate bath to keep its temperature high or low according to our guidelines.
We ran each of the 4 spec tubes for 10 minutes all at the same time and took readings approximately every minute. These are the results that we obtained.
We can see the data here presented in a tabular form, Table 1. We then transform this data into a line graph. Our hypothesis is concerned with the initial rate of reaction of the enzymes, so we had to look at the rate of reactions for each of the enzymes. Now the line graph shows the percent absorbance over time of each of the 4 test tubes. Since our hypothesis was concerned with the initial rate of reaction, we looked at the first 2 minutes of what happened for each test tube. Now to find the rate of reaction, we took the percent absorbance at the 2-minute mark and subtracted it from the initial percent absorbance at the 0-minute mark. We then took this value, divided it by 2 to get the initial rate of reaction over the 2-minute interval. Now this data was then compiled into a new table, Table 2.
This graph shows each rate of reaction for each of the 4 test tubes. As you can see, the higher the temperature, the higher the rate of reaction.
The entire system. We repeat, this is only a test. This is only a test. The mind, the body, the positive, the negative, the ground.
In some experimental trials, the 4th test tube showed a decrease in percent absorbance. This may be due to denatured enzymes or lack of substrate or some confounding variable. When the temperature is higher, the molecules have a higher average kinetic energy. This means that the substrate can move faster and has a greater chance of binding to the enzyme. When the enzyme is bound to the substrate, it can go ahead and catalyze the reaction and therefore increase the overall rate of reaction. Each enzyme has its own optimum temperature. The optimum temperature is when the rate of reaction is highest for that enzyme. For our enzyme, catechol, the optimum temperature could not be concluded from the data provided. The rate of reaction increased as temperature increased. We never could conclude where the rate of reaction was highest. Now, some implications can be concluded from the data of our experiment. For example, a perfect model would be our human body. When we have a fever, our body is raising the temperature of our body. Now, the temperature is being raised in order to activate the white blood cells, enzymes, so that they can do a better job of killing off bacteria and viruses.